Hello, welcome to another session of VerEvMed, where friendly scientists answer your questions. Today we're going to talk about glioblastoma megaforma. We're going to talk about drug and radiotherapy resistance via corruption of ceramide-mediated tumor cell apoptosis. My name is Dr. Dan Guerra. What is GBM? It's the most prevalent diagnosed primary adult malignant brain tumor. After onset, the survival is really poor, less than 10% by 2 years. Precise etiology of GBM is complex, variable, and uncertain. There are 3 highly correlated GBM-linked biochemical pathways that have emerged, however. One involves a tumor suppressor p53, which of course is a common mutated gene in many cancers. Another is the tumor suppressor RB, the retinoblastoma protein, which is also relatively common. Both those proteins are checkpoint proteins in the cell cycle. The third pathway that has been linked to GBM is the receptor tyrosine kinase, phosphatidylinositol 3 kinase cascade system, which is also often found in cancers. Besides those 3 systems, there have also been reports of alterations in lipid metabolism and, of course, apoptosis. And since those have been linked to drug therapy refractory GBM, the paper I'm going to talk about today was the research was conducted. Here's the paper. It's going to be talking about ceramide synthase and another protein called BCI2L13. And I'm going to call this an unforgiving association, which leads to drug therapy resistant GBM, tumorigenesis via mitochondrial anti-apoptosis mechanism. Paper is from Proceeding National Academy of Sciences that came out about 2 years ago, 2014, and the first author is Jensen. As an introduction, what is ceramide? It's essential for mitochondrial apoptosis progression. Of course, it's a lipid. De novo synthesis of ceramides is catalyzed by an enzyme called ceramide synthases, and there are 6 different isoforms of that. And they differ in what fatty acid is attached. That's what that enzyme does. Makes an avid linkage to a sphingosine base. Apoptosis itself induces ceramide synthase on mitochondrial-associated membranes called MAMs, and that compartment links to the ER directly, a physical linkage, ER to the mitochondria. Ceramide production is therefore necessary for conventional, or that is, normal physiological BC12-associated X protein insertion into the mitochondrial membrane. These oligomerize, they form a pore, and that causes the outer membrane protein of the mitochondria to permeabilize, that's called MUMP, and then cytochrome C is released, and you get the throughput, the follow-through of apoptosis. I told you we talk a little bit about ceramide being a lipid. It's synthesized from palmitoyl-CoA and the amino acid serine. Once you make that keto product, you then reduce it and you make sphingosine, shown there on the right. And then the rest of the reaction involving ceramide synthase is shown here. Don't worry about what DOX is. This is from another paper, but that's doxorubicin actually. But you see here how sphingosine then combines with the fatty acyl-CoA to make ceramide. Notice that ceramide directly is linked to apoptosis, and also note that ceramide can be used for other more complex sphingolipid metabolic pathways. Here is ceramide, so that red linkage down there, that's a fatty acid, and that can be polyunsaturated very long chain, or it can be monounsaturated very long chain, depending on the molecular species of ceramide synthase that synthesize that amide linkage to that fatty acid. So ceramide synthase 2 is pro-apoptotic, therefore it enhances chemotherapy and it tanks tumor growth. So this is all a positive outcome that you'd want by doing chemotherapy for a disease like GBM. In fact, low expression of ceramide synthase 2 is a negative indicator in breast cancer, so it's been studied not just in GBM. the isoform 6 promotes therapy-induced apoptosis in colon cancer, head and neck squamous cell carcinomas, and in lung carcinomas. So it's got quite a reputation of being a positive player, positive agent in cancer remission. Current paper again comes from PNAS, demonstrates that this other protein, BC12L13, actually inhibits that ceramide synthase, therefore inhibits the production of ceramide. And in so doing, it elevates glioblastoma and other solid and systemic bloodborne and metastatic human cancers. This particular figure here comes directly from the paper. At the top there, you can see an A, you can see that in ALCL, AML, and in GBM, okay, these two blood cancers, and there's GBM, you get an elevation of this unique form of this BCL protein. See much higher levels of it. This is an in silico examination of the amount of the protein. And then when you look just at GBM, you actually get a 100-fold increase. This is at the QRTP PCR levels. This is looking at transcript level. You're looking at a Western blot here in C, and you'll see this is a higher level of that protein, that bad player protein, the BCL protein, in all of these tumors shown here, but not in the non-tumorigenic cell mass that was analyzed. And this is that protein that is quantified here. This is just showing the tissue distribution of that BCL protein. And here you can see the protein is quantified from that Western blot. And again, you get about a 100-fold increase in that bad player protein. So obviously it's involved. The conclusions of this paper, and we didn't show you a lot of other data, but basically let me just cut to the chase. BCL2L13 contributes to therapeutic resistance in cancer and directly corrupts the ceramide pathway. It physically binds to the ceramide synthase 2 and 6 via a specific amino acid sequence called BHNO. That's a domain. And there, because it does that, it inhibits both homo and heterodimerization of that ceramide synthase. When it does that, it inhibits the activity of the enzyme. So that domain encompasses about 250 amino acid sequence in that polypeptide. Obviously, that would be a possible druggable target. Just in a more general summary, apoptosis is a mechanism for GBM remission. Ceramide promotes apoptosis. Overexpression of the bad player BC12L13 in GBM blocks apoptosis by preventing ceramide synthase, therefore causes the progression of GBM. New drugs that maybe target that system could aid in GBM therapy. Okay, just to give you another idea about this, has also been looked at in other systems. Here's a more recent paper in Neuroscience Oncology, it's 2016, showing you that acid ceraminidase confers radio resistance to glioblastoma. So the acid ceraminidase is an enzyme that breaks down ceramide. So if you break down ceramide, it's the other end of the spectrum, right? Rather than inhibit its activity, then you get resistance to radioisotopic treatment, therapy for GBM. So glioblastoma is a brain cancer in glial cells. It's pretty prevalent. You can see there are some 70,000 new cases in 2013. It's the second leading cause of cancer-related deaths in children, both sexes, under the age of 20. So it's quite a nasty player. Leukemia, of course, is first in that regard. It's the second leading cause of cancer-related deaths in men in that age group. And it's the fifth leading cause of all cancer-related deaths in females of that age group. Our email address is shown there. There's our website. Remember that we are scientists verifying published evidence in medical biosciences. Thank you very much for your attention. Again, my name is Dan Guerra, and it's been a pleasure talking to you today.